Good morning to all and welcome to this presentation. My name is Steve Labrie. I am a professor at the Department of Food Sciences at Laval University, and I'm a director of the Stella Dairy Research Center. This morning, my presentation is a presentation that covers all of my research, the research that I did within the context of projects financed by Novalait, but also financed by other sources. And the presentation will focus on the natural microbiota of cheese. This morning, I will talk to you about the importance of natural microbiota. What is its importance in an impact on cheese? I will talk to you about work on natural microbiota, the characterization of some microorganisms that we have identified in the natural microbiota of cheese, and deal with the impact on typicity of cheese. And finally, I will finish my presentation with new questions that we have generated in our research. Obviously, we do research. Often more questions come up than we have answers to. And that is what this portion of the presentation will bring us. First of all, let's begin by talking about the importance of the natural microbiota. Cheese has a very complex ecosystem. If we take a cheese such as this one here on the screen, and if we look at this in a closer manner, if we look at the surface and the heart of the cheese, we realize that there are microorganisms that have developed within and at the surface of this cheese. And it's even more clear for specialty cheeses. So on these images, you can see that we have the mycelium that has grown at the surface of this type of cheese. If we now look at electronic microscopy, we can notice a number of microorganisms. In this slide here, you can identify bacteria, we can identify yeast and filaments, mold filaments. The ecosystem interacts, the microorganisms interact amongst themselves, between themselves, or with the matrix, and will eventually have behaviors that are either called cooperation, or which means that the cheese will develop in a favorable way or an unfavorable way. In the cheese ecosystem, there are two big categories of microorganisms. The first group involves microorganisms that we use on farms. So microorganisms that we will inoculate voluntarily to milk to make cheese, and which is made up of different groups of microorganisms. Here we have bacteria, yeast, and mold. Based on the variety of cheese, obviously, we will use one or the other, or a combination. In the second group, we're talking about natural microflora, which is naturally occurring in cheese and milk, involving bacteria, yeast, mold, and can also contain bacteriophages, which could have a negative impact on ripening or production of cheese. For the natural microbiota, we see a number of types of bacterial species, and some of them can have some functions which differ depending on the species. And we can find microorganisms that are favorable, others that are unfavorable for cheese production, and others that lead to favorable or unfavorable characteristics, and some are even bad for consumers. So we're talking about the good, the bad, and the ugly. All of this has an impact on the evolution of microflora as present. Indeed, over time, there are some microorganisms that will progress and turn their roll over to other microorganisms, which will find the new conditions as being favorable to their growth. This means that in some types of cheese, we can see some significant changes in microflora as present in the product, and this can reach 90% of modification between initial microflora and final microflora, which is present in the product. In addition to this, this evolution will lead to olfactive and taste notes that are different, and microorganisms will have an impact on the sensory profile of cheese. Natural microflora that develops, given that we control it more or less, can lead to variations between different types. So in some conditions, based on the matrix and the presence of microorganisms in the environment, variations around the quality of batches can be observed. And finally, natural microflora can also be a typicity element in cheese. Some species will have notes, organoleptic notes, that could be typical of a region or a production, given that these microorganisms are very typical and represent the region or the cheese plant where the product is made. So our work on natural microbiota began more than 15 years ago now, and we have focused on mold sections and specialty microflora. Our work began on fungal microbiota present in milk from Quebec. We recruited a number of processing plants and farms, and we checked the presence of microflora, fungal microflora in different samples. The goal of this project at the time was to establish what fungal species were dominant in Quebec milk, and we wanted to map the species that were present in different locations. We also wanted to verify an assumption to ensure that the fungal microbiota was stable in time or if it varied based on time. We also wanted to know whether there were certain things that could influence. So yeast, for example, present in milk, could that survive through the process of processing, and if it could find its way into cheese and eventually modify or alter or participate in the ripening of cheeses. This led to one of the very first publications by our lab and one of the most quoted publications. This is the characterization of the fungal microflora in raw milk in specialty products and cheeses in Quebec. In this study, we checked yeast and mold presence. So we established a ratio between both groups of microorganisms for 21 processing plants and cheeses and farms in Quebec. For 4 of these, we checked the fine composition in yeast species, as you see here on the screen, and the molds down here at the bottom. We determined for the plants that there was a certain level of consistency in terms of the presence of yeast and mold. And in this case here, we realized that the presence of yeast was higher than that of mold. In other cases, like here, we could also see that the total content in fungal species, so containing yeast and molds, could be fairly low, and therefore it's less high than the plant that we saw earlier, and that the composition could be different as well. So just based on the names presented here around yeasts and molds, we have species that are different. And here, just with the species that are present here, a number of these favorable yeasts to ripening are present, which is a good indicator for this cheese plant. Our results for this study were therefore among the farms studied. We had 3/4 of producers who displayed dominance in yeast, while 1/4 of producers had a light dominance in molds. In terms of the cheese plant, often we want to have yeast dominance rather than mold dominance, since molds can be contaminants that can develop and alter products. It remains that a few species were also unique and unique to some farms for a reason that we don't really know right now. We had some species that were found for one or several farms, and even these species were unique and dominant in the microflora of farms. The idea is now to understand what the role of these species is, and the species identified, could they play a role or have an impact on different types of cheese. One of the spin-offs of this project was the use of project results in the specifications, leading to what we call the Canadian cow cheeses. In these specifications, we were able to use the results of our study to show the difference between fermentability in Canadian cow cheese milk and milk from other species. So this work was done with my colleague Danielle Saint-Gelais at the CRDSH at Agriculture Canada, and the chemical aspect of the characterization of the Quebec milk and different microbiological approaches made some differences. So for Canadian cows, there are a lot of other arguments. I would not want to limit specificity to those studies that we did only, but we are proudly knowing that our results can contribute to some specificities as indicated here, and we are proudly announcing the results of our study. Once milk characterization is done, we then were interested in the volatile compound aspects that we isolated. Some yeasts seemed to be interesting, and we wanted to determine their potential for the ripening of cheese. What you see on the screen currently is an analysis profile. So we have our volatile compound profile for 2 years and of 2 yeasts, and we put into our device some fermented curd with 2 different yeasts. For each of them, you have the profile which is presented on the screen. And for we have the black profile and the green profile, so Orientalis and Cerevisiae. What this allows us to do is to eventually determine for each of the peaks presented which component is present and how they contribute to cheese ripening. In this case here, we will have components with different flavors, smells, whether they be burn, rancid, green, oily, fruit, or other descriptors that could be present in the taste of cheese for the different peaks that are observed. So for some of these descriptions, it could seem not particularly tasty, but it's the combination of each of these peaks and components that makes up the complexity of the flavor of a cheese. And the important part here is to identify which contributions of these microorganisms or have high flavor potential. More recently, we followed the development of the ripening of different types of cheese, and specifically we developed follow-up methods that use metagenomics, as I presented in the last slide, for 29 specialty cheeses from 22 different cheese plants. Again, we identified different microorganisms that were present in these ecosystems, and we sequenced more than some 20 genomes from interesting microorganisms, and we are continuing to analyze the genomes And we are in the process of looking at different tests so that we can look at the contribution of microorganisms in ripening. We are also determining the activity from some strains on cheese quality, very specifically texture. So we accumulated a lot of results, and we are looking to present a number of articles in the next few months looking at the information that we gathered during our testing. As I mentioned from the get-go, our studies focus on cheese ecosystems. So you can say, yes, well, the cheese ecosystem is more than just isolating microorganisms in an individual way and checking their activity. You have to see that as a whole with interactions between the different microorganisms. In our lab, we've begun working on metagenomic methods that allow us to consider the ecosystem overall. Metagenomics, depending on methods and approaches used, allows us to answer a number of questions, including the question, what is present in the ecosystem, whether it be eukaryotes or bacteria, and then the question of who does what. So who is there? What are they doing? In terms of metabolic pathways, appropriately, we can determine what the metabolic pathways are as they're present, and then to see what products from this metabolism can contribute to cheese ripening. And metagenomics also allows us to answer the question of how these microorganisms can contribute to ripening. Here I'd like to show you a metagenomics process that was used in one of our projects. And in this case, if we wanted to check what the microflora was that was present at the surface or within a cheese, by metagenomics, we can see the microbial diversity that is present. To be able to accomplish this study, we totally extracted a DNA that was present on a given well-calculated surface of a cheese. And we checked this for a number of places on the cheese. And again, once the DNA was extracted in this DNA bubble on the screen, there's the presence of DNA of all microorganisms in the sample. And when we use a sequencing method, then we can generate a large quantity of sequences that come from this DNA. Once an organism is available in the extracted DNA, the more we are going to sequence it at a number of frequent times. This allows us to understand species proportions in the profiles generated. So the more things are detected, then the more this bar for more microorganisms will be based on the profile, thus allowing us to look at the different profiles obtained for different cheeses or different conditions. We used this approach for 49 cheeses coming from 21 cheese plants, and the analysis was done in 2015 and 2018. The idea was to look at the evolution of microbiota in between these 2 years. We looked at biology. We were able to do mapping of different cheese microbiota, and we were able to assess the significant changes between cheeses, periods, and identifying microorganisms that are very specific for the cheeses in question. Here, I'd like to show you what this looks like. So clearly there are a lot of data in these charts. On the left-hand side, you've got the abundance of bacteria in different cheeses from the Quebec territory. And in the right-hand portion, you have the fungal microorganisms in these same cheeses. We did the same analysis for the center of the inner part of the cheese, but also for the rind, bacteria, and fungus. We also categorize cheeses according to what they look like. So if we had natural rind, washed rind, or other types of rind cheese. The study allowed us to determine the similarities, but also the differences between different cheeses that were analyzed. It also allowed us to identify more specifically species that could be interesting when it came to cheese ripening. And following the study, we also rendered a personalized report available to each of the cheese plants that participated. So their microbiota in 2015 and their microbiota in 2018, and their differences according to the average in the same category of cheese. So the study allows us to better understand the distribution of species through artisan cheese shops in Quebec, or cheese plants in Quebec, and to identify the main contributors to ripening. As I said from the get-go, obviously research often leads us to asking other questions to be able to continue our understanding. New questions that are brought forth by our project are the following. So who is absolutely essential in the ecosystem? What surprised us to a certain extent when we looked at cheese ecosystems was the great diversity of species that were available in the microflora of products. Even if I'm a microbiologist and have been so for some 20 years now, I did notice that there were a number of species in the cheese ecosystem that we didn't even imagine could be present and which could potentially contribute to cheese ripening. So right now, all of these new species, so the fungal species that were identified, specifically, that allows us to determine whether they are really important for ripening and what their contribution to the ecosystem is. Another question that we are asking is who does what exactly? So along these same lines of who is important, well, what is the real contribution to ripening? In the third question, so what are the sources of these microorganisms? We know that they are present in cheese and they develop, but are they now microorganisms that are specifically in line with the cheese? Do they come from the farm? And are there ways of favoring the emergence of these species of interest in products? So these are the questions that we will address in the next studies that we expect to undertake. In addition to this, how do we maintain the environment of the cheese plants, and how do we maintain interest to ensure that we already have species in the environment to contribute to cheese ripening? But in addition to that, how do we move forward so that we can retain these within the walls of the cheese plant without spreading them? If ever there was a problem around balance, the microbiota, whether it be bacteria or fungal in the plants, how can we reintroduce species that are of interest to us in the process to ensure quality ripening? So these are the big questions that we need to answer. And again, with a number of years of study, we can continue to go over all of these questions. First of all, in order to continue our research, we have projects ongoing that are financed by the Dairy Research Cluster Tree and financed by Dairy Farmers of Canada, Novalait, and Agriculture and Agri-Food Canada. In this project, we have the opportunity of counting on a multidisciplinary team that is extremely dynamic, and my colleagues Claude Champagne, Sylvie Turgeon, Sylvie Turgeon, Martin Breton, Yves Poliot, who has retired but is one of the project mentors, and who were added. And then we had Danielle Saint-Gervais, Julien Chamberlain, and Marie-Claude. And I would like to now thank all of the organizations who supported research in our lab and have done so since the beginning. So specifically Novalait, they help us tremendously in the study of the natural microbiota in cheese and milk. And I would like to highlight the contribution of the 27 cheese plants that provided us with cheese and milk and who have done so since the beginning of our studies. So these are contributors who are extremely enthusiastic and with whom it's a pleasure to work. So thank you very much to the cheese plants and shops who participated. Again, I keep their contribution confidential, but do know that we have much appreciation for all the work that they do for us and the samples that they supply. In addition, I would like to thank my research team. Here you have some photos. More recently, we've been in lockdown, but COVID-19 clearly has affected our research work and our way of working. But I've got an incredible team. And even if students continue to come through, it's always a pleasure of working with all students. Thank you very much for your attention. I am available for the question and answer period as that will follow, and do not hesitate to contact me. I will always be pleased to answer all your questions, whether it be during the conference or during other times. So if you want to contact me by phone or by email, I'd be happy to talk to you. So thank you very much for your attention.